Hello and welcome to this webinar titled Platform Technology Development for Biosimilar Upscaling, hosted by Biopharma Asia Magazine and presented by Dr. H. Poon, President at QuaCell Biotech Limited, and Floris de Smet, Technical Director, Bioprocess Solutions at Sartorius Stedim Biotech, China. My name is Orsolya Balog and I will be the moderator. Now, please allow me to introduce our first presenter, Dr. H. Poon, President at QuaCell Biotech Limited. Dr. Poon is currently the President of QuaCell Biotech Limited. He previously worked for Hyson Biopharm, Merck, Roskamp Institute in the USA. He completed his PhD in biological chemistry at University of Kentucky and MBA from University of South Florida. He has years of experience in the field of biological therapeutics, analytical research, bioinformatics, and proteomics. He has more than 10 years of cell culture experience with focus on biopharmaceutical products. He published more than 30 peer-reviewed articles and contributes to more than 5 book chapters. He is currently the managing editor for Biomedical and Pharmacology Journal, associate editor for Journal of Chemistry, Biochemistry, and Molecular Biology, Banned Journal of Bioinformatics and Biometrics. Research interest: biotherapeutics, proteomics, metabolomics, cell line engineering, cell cultures, multivariate analysis, QBD, and PAT. Welcome, Dr. Poon.
Thank you for the great introduction. So today, I'm very happy to be here today, and today I will be maybe having a discussion with everyone regarding to the platform technology for biosimilar upscaling. And I think mostly I would be focused on the cell culture aspect of the biosimilar. Okay. Hold on a second here. The slide is moving by itself. Okay, just give me a second. Are we still on the first slide? Excuse me, the slide is moving by itself. Hello?
Hello, I think we got a little technical problem. Would you like to start your presentation with the first slide, Dr. Poon?
Yeah, the slide is moving by itself.
Oh, okay. Just bear with me.
Okay. All right, I think I'm at the first slide now. Okay. Let's see here. So essentially, I will start with the slides is moving. Okay, so my talk is divided into 3 parts. The first part, I give you a little bit of background of QuaCell company. And then the second part is kind of review on the platform technology in the current market. And then at the end, I'll give a kind of like a case study regarding to the how do we apply the platform technology for scale up. And so QuaCell is a new company, and our focus is in cell culture media. But we also do a lot of the cell line engineering, as well as the process development, as well as process characterization. And so some of our products is QuaCell media, Hex 293 cell culture media, and hydrosomal cells media. We mostly focus on the CD media and also for suspensions. And some of the service that we provide, like I said earlier, we do cell line engineering, media development, process development, characterization, as well as the custom media manufacturing. Let's see. So just a quick review on the media development, biosimilar media platform. Okay. This is not working. The slides is going by itself.
Okay. I do apologize. Dr. Poon, I will get in touch with the technical people, and we will solve this problem as soon as possible.
Let's see here. Let me just go back to, okay, maybe I'll just go ahead and, so the next part is kind of describe the current platform technology. And when we talk about biosimilar platform, so essentially three platforms are particularly important. The first one is the cell line platform, and the second one is the cell culture media platform, and the third one is the bioreactor platform. So that's more related to the process. And so for the cell culture platform, just give me a second here. So for the cell culture platform, essentially right now there is the two main selection platform was the DHFR as well as GS. And of course, CHO-K1 is also a very common cell line to be used as a wholesale for the biosimilar. But in order to increase the titer, the selection using DHFR and GS system usually is more preferred. And currently, there's a couple companies they are selling the GS system, but because a lot of the biosimilar developer, they are cost is a really major concern. So the licensing fee, it really is a big issue for the biosimilar developer. And the second part for the cell culture media platform, a lot of the cell culture media platform, people are using the commercial media. So essentially it's a catalog product. And on my slide, which you can see in here, I actually give some of the different companies' products, the catalog products on there. And you can see as the first generation of the product. And that usually supports somewhere between 1 gram of the product. But the second and the third generation, this platform media, they actually become more and more able to increase the titer of a lot of the biosimilar molecules. And for some of the third generation products, they are able to go to up to 4 gram or 5 gram per liter. So as you can see, in the last 10 years or so, the media development have really increased and make the titer become higher and higher. And the last one is the bioreactor platform. When I say bioreactors, mostly I'm really focusing on the high throughput, the microbioreactor as well as the miniature bioreactors. The reason that is such an important platform is that because when people are developing biosimilars, one of the things that is important for them was to adjust the protein quality similar to the reference product or the originator. And so in many cases, you need a bioreactor system that can screen many media as well as a lot of the bioprocess for you when you're doing the screening. And so that's why some of the high throughput screening is important in biosimilar products. And so on the market, there's like what I call the micro bioreactors. So those are usually less than 100 milliliter, and those are very good for screening clones, media, as well as the some of the process parameters. And then the miniature really gives you a good way to scale up, so that you can kind of have the process develop. And so the remaining part of the talk, I'll be focusing on one of the scale-up platform that we developed. So when you're looking at scale-up or maybe scale-down, scale-up platform or scale-down model, you actually, a lot of it is really a physical model. So if you look at, for example, KLA, the mix time, sometimes people use computer fluid dynamics models, CFD. So all this really is a basic way to look at a scale-up model. So for us, we want to look at it a little bit differently. We want to really looking at the biological model. In other words, we want to look at how does the cell metabolize, how the metabolism is changed during the scale-up process, and can we use it as a way to scale up. So our focus really is on the biological aspect of the cells. And so, so in order to test out this theory, we actually use a very simple project goal. We really just want to look at the one of the protein quality, and then as this cell scale up, we look at the metabolism of these cells, and then to make sure it is equivalent in the small scale versus the large scale, and therefore we can qualify the small scale as a small scale down model. Also, that we can use this model for us to scale up. And so what we do is that we essentially run a lot of small scale bioreactors, and then we use this to build a mathematical model, and then using the data that we obtain from a larger scale, and then we put it back into the model to see if there's a statistical significance between the small scale and then the large scale. And then if there's no statistical significance, we will be using that as the model for a scale down, as well as using it for a scale up. And so the metabolites that we're looking at are relatively simple. We just look at glucose, carbon dioxide, lactate, also some of the ions. So those are as the input into the model. And the output is we're using titer, VCP, as well as the variability. And so the reason we picked that was because this is our first try, first experiment. We do not want to complicate it with so many inputs, so that it will be easier to understand, so that we can have a proof of concept to see using the metabolites to set up a model if it is possible or not. And so this slide showed that this is some of the data that we generated in this in the small scale of the 2-liter and 30-liter. This is different titer as well as the different maximum viable cell density. And so with all this data, we put it into a multivariate analysis software called SIMCA. And the SIMCA will generate a mathematical model for us. And by looking at the model, we determine that 7 principal components that is significant for this model. In other words, there's 7 major factors that is affecting this model. And so the next step we do is we really want to see if the small scale model really comparable to each other. And so what we do is we plot the data into a batch mode, and therefore we see that all this data are within 3 standard deviations. And therefore, 99% of the time, the small scale will be similar to each other. And then we also use the software to really examine to see when we're doing the large scale, is that different between the small scale and the large scale. And the statistic display that we used was using Hollingtow's T2. And what we were able to see is that all this bioreactor runs was within our population. In other words, within 95% confidence that all these are similar to each other. And so after we were able to scale down, we decided to look at some of the metabolic changes. So we actually do a metabolic study within cells, and then using all the metabolites that we saw changes from large scale and small scale, we are actually able to see some of the metabolites that is associated with the high pH changes and temperature changes, and also the titer, or how's the metabolite related to the titer. And so, with all these changes, we are able to build a model that is, we are able to qualify the model that is comparable in 200 liter versus a 2 liter. And also, by doing this process, we're able to have gain significant knowledge of our process. In other words, what metabolite is related to the critical process parameter as well as the critical quality attributes. And we also identify one of the metabolites as a biomarker for us to use it as a PAT to monitor the IUC, but we actually did not show detail on this particular metabolite. And then at the end, we're able to establish a metabolic profile which we can use to help us to scale up. And this metabolite model can be used for scale up, and also, like I mentioned earlier, we use this metabolite to qualify our scale down model by a statistical analysis through multivariate analysis. And so that's all I have for today.
Thank you so much, Dr. Poon. Once again, I would like to remind our viewers that there will be a live question and answer session at the end of the presentation. Please feel free to send in your questions at any time during the presentation via the Questions tab located at the right-hand side of the screen, and we will go through these at the end of the presentation. Now, please allow me to introduce our second speaker, Floris de Smet, Technical Director, Bioprocess Solutions at Sartorius Stedim Biotech, China. Floris de Smet has over 10 years of experience in the bioprocessing industry. He has been with Sartorius since 2007 in various sales and marketing positions in Europe, US, and Asia, including global key account management, PD consultancy, and strategic marketing. In his current tenure, he leads the field team of application specialists and marketing managers in China. His topics of interest include molecule lifecycle management, alliance management, corporate business development, and strategic marketing. Floris holds an MSc and M.Econ both from Ghent University, Belgium. Welcome, Floris.
Thank you very much, Orsalia. Let me load my slides. I hope I'll be more lucky than Dr. Poon. Okay, bear with me, please. Okay, I think we should be good to go. Good. Good morning, good afternoon, everyone. Good afternoon from China. Thank you, Orsalia, for the kind introduction, and thank you, Dr. Poon, for the nice first part of the presentation. Unfortunately, some of the slides fell off, but yeah, I hope you can find a way to still reach the audience with the first part of your slides as well, because I think it's a very good story, which I will continue to build upon in my part of this talk now. I will effectively talk about the points that were listed as the learning goals of this webinar, and we'll be talking about some of the tools in the context of some of the typical projects that Dr. Poon presented as well, to help candidates move quickly through development, to build processes from there that are low in variability and robust, and we'll be strongly relying on the concept of high throughput bioreactors in my overall storyline. Okay. Good. The reason why I think this is an exciting talk is specifically in the context of biosimilars. Technology choices really impact the success of an overall biosimilar business strategy. So I think when we think of making biosimilar strategies successful, looking at the life cycle of your molecule and how technologies can help your life cycle to be successful is extremely important. Thinking of critical success factors to launch biosimilars on the market, I would say that most of those are really directly impacted by the choices that one makes in the technology space. So it starts with selecting the right candidates, meaning if you have the technical feasibility to produce those candidates at the right quality, and then finding ways to develop those and bring them faster on the market than your competitors, and once they are there, being able to manufacture those at the lowest cost of goods, and of course, in the process, being able to build in the right quality and fingerprint-like similarity between the innovator and the biosimilar. So when we then link this to what this means for technology, is that it is important that we don't just look at technology in the research space, serving the research group, or technology in the PD space, serving the development group, or the manufacturing platform, serving the MSAT groups or manufacturing suites, but that we really think of an aligned platform that starts in R&D and that hands off a developable candidate to PD, where the platform in PD can give a robust platform that will consistently yield the products at the right quality, and do so cost efficiently and quickly. So the paradigm, I think, for the technology choices and the integration of technologies is very much to hit that sweet spot in speed, cost, and quality when it comes to being successful in a biosimilar business strategy. The thesis of my talk today is then also that Sartorius has such a complete and integrated platform, specifically in the space of upstream development and upstream technologies. And it very much relies on the concepts of high throughput screening tools for cell line and media development, process development, and process characterization, with scaling concepts from those high throughput bioreactors to production scale bioreactors, together with advanced analytics and software packages to help scale those steps over the molecule over the different steps of the life cycle in a seamless way. So by using the high throughput screening tools that I will be talking about and seamless scalability concepts, we can really drastically reduce the time to clinic and the time to market. Before we start talking on the technology side, I also would like to highlight the service package that Sartorius can offer today. Since a few years, we also have now a complete solution for cell line development, cell bank manufacturing, biosafety testing, and analytical services. I'll mainly focus in this talk on the analytical services. On the cell line side, I just would like to say it's a CHO DG44 cell line platform that we offer that gives 3 grams per liter titer in 95% of the cases, 3 grams per liter or higher, and gets you to your research cell bank within 4 months. But for the sake of time, I will mainly focus now in this talk on the analytical testing services, which is a service that we offer from our BioOutsource affiliates, which we acquired a few years ago. What they offer is ICH compliant testing, specifically for structural testing of antibodies or recombinant proteins, antibody binding testing, and functional testing of biosimilar molecules against the innovator molecules. So this fits perfectly in the structure binding function paradigm, or the totality of evidence that the regulatory agencies expect when bringing a biosimilar on the market and demonstrating the fingerprint-like similarity. The interesting point about BioOutsource is that they have developed, by heavily focusing on these analytical testing services for biosimilars, they have developed a full set of pre-qualified and pre-validated off-the-shelf assays for characterization and compatibility testing of most of the hot biosimilars that are currently being developed or are already on the market. In addition, they are also working, or we are also working on providing those same types of assays for the subsequent waves of biosimilars that are expected to hit the market in the next few years as well. So this is, of course, a service that drastically reduces method development time and method transfer time, and gives you the right tools to make sure that you can effectively demonstrate that your biosimilar will be produced and will be launched on the market and produced on the market at the right CQAs. When we then move on to the technology part, I want to start focusing on the high throughput screening platforms. So the first high throughput screening platform that I would like to introduce is the Intelliside Iq Screener, which is a high content, high throughput flow cytometry technology that is coupled with high content imaging. What the technology does, it is a technology that will be profiling and screening individual cells in suspension for phenotypic and functional endpoints. For instance, target expression, antibody binding, cell death, and so on. And it's working based on high throughput screening, antibodies and dyes, and multi-well plates together then with the high content software. This technology mostly finds its use in discovery applications. However, because of what it does, screening the behavior of cells in suspension can also be used for the early stages of cell line development, for the screening of the clones that picked up the genetic material to see how productive these different clones that picked up the genetic material are, and how suitable those would be for industrialization purposes. Typically, when people look at clones and selecting the right clones, people will be looking at titer. So selecting the clones that produce the antibody with the highest titer. However, this can be deceptive or misleading because you don't really know anything about the productivity per cell. So what the Intelliside Iq Screener will do is, because of the high content, high throughput screening platform, it will not just give you an overall indication of the titer, but it will also give you an indication of the productivity per cell and the productivity per viable cell, which means that if you combine all those readouts, in addition to the overall productivity, looking also at the cell density, the IgG per cell, and the cell viability, that you would probably choose different cells with higher cell-specific productivity than those that you would select if you would only look at the titer. So which helps you to drastically reduce your clones that picked up genetic material to only a handful that you want to select for further development then. Moving on from there, once you have that handful or half a dozen of clones that you want to push through development, then we move to the AMBER 15 high throughput screening system, which is basically a high throughput screening bioreactor platform, an automated workstation that will run 24 to 48 15 milliliter bioreactors in parallel with automatic feeding, automatic sampling, and so on and so forth, measurement and control of pH and DO to be used typically in applications of cell line development and media development. So the idea is here that you would go from the typical way of using high amounts of shake flasks to a more automated way of working using the AMBER 15 high throughput automated workstation, which doesn't only save in time, but because of the way that the bioreactor looks like, actually it is a bioreactor-like condition with an actual sparger and impeller in it. Because of the way it looks like and because of the way that it is designed, it also mimics the industrial application in a better way than the typical shake flask. So what we see in these graphs is that when you look at the comparison of CQA profiles between AMBER, a 2-liter benchtop bioreactor, and a shake flask, is that sometimes there can be some deviation in what we would see between the shake flask and the 2-liter bioreactor, but the higher consistency between the AMBER and the 2-liter bioreactor. This is specifically prevalent in this graph where we look at the glycosylation. This is a case study from Monza Biologics where they screened 10 CHO cell lines for specific productivity and ranked them in order of specific productivity, and in parallel did the same experiment with a 10-liter benchtop bioreactor. And we see that the ranking of the AMBER 15 results is exactly the same as the ranking that you would get with a 10-liter benchtop bioreactor, proving the point that the industrial design or the industrial-like design of the AMBER 15 significantly helps in helping us to select the right clones that will be able to produce with the desired productivity in the industrial scale applications as well. Here is another example, this time by Cobra Biologics, which used the AMBER 15 to screen 8 CHO clones to basically identify the best clone and the best feeding strategy. So running 3 clones in parallel with 3 feeding strategies each, and at the same time assessing the clone stability in this experiment. So devising a multi-parametric set of experiments. And if you look at the way they did this and the time they saved by using the AMBER 15 high throughput automated workstation, they were basically able to save 16 weeks of development compared to a 4x5 liter traditional benchtop setup. So 16 weeks translating to 4 months, certainly is significant when you think of the criticality of bringing biosimilars to the market quickly. So the AMBER 15 will allow you to increase the efficiency, reduce the timelines, which helps you to not only make sure that because of the industrial design, you will select the productive clones that will then also be productive in process development and the industrial applications in scale-up, but also it helps you to speed up early process development or do more experiment with the same resources. Moving on then to the I would say the process development and the actual the steps in the process design, where we really start to look at the industrial considerations for bioreactor in detail. I would say that the left part of this slide is the representation of what has been considered in the industry for decades as the benchmark and as the gold standard for producing recombinant proteins in a variety of different applications. The vertical shaft, vertical stir tank with a 90-degree angle, and this is exactly what we also have built in the Sartorius single-use stir tank bioreactor design, because we believe that it is important that we leverage the proven efficiency of the gold standard in the stainless steel design to a new single-use standard that then can also leverage the same efficiency in the new processes deploying single-use technologies. The reason why that is important is because the traditional design that I just presented finds the optimal spots in the design space in terms of finding the right spots in terms of having a power input that is not too low and not too high, and a gas flow that is not too low or not too high, meaning that it is a design that helps you to find the right balance in your parameters, combining high efficiency and making sure that you hit the optimal productivity, and at the same time making sure that the risk of your process and the chance for your process to be safe and reproducible is higher. And this is really thanks to the classical stirrer design that we have built in the single-use bioreactor range and the versatile gassing concept that we have built in the biostat STR, the different biostat STR scales. If we look at how this now plays out in the different scales, I mentioned that we have built in this classical stirrer design in the single-use stir tank larger scale bioreactors, but of course this has already been present in the benchtop bioreactors, which Sartorius has been bringing on the market for many decades. And we see here that the ratio of the vessel height to the vessel diameter and the ratio of the impellers to the ratio of the vessel, the diameter of the impellers to the diameter of the vessel, 1.8 and 0.4 consistently across scales respectively. However, we also see, interestingly, that those ratios are exactly the same in the Amber's 250 product range. So that means that we have a consistency in geometry in the bioreactor range from the 250 ml scale all the way to the 2,000 liter scale. Meaning maintaining this geometrical similarity in vessel design and keeping the mixing and gassing strategies consistent across the scales definitely will help to simplify scale up and scale down studies, helps you even to switch between conventional stainless steel and single-use bioreactors. Single-use may not always be the best choice for every single process. One really needs to run the economics given the estimated output of your plant. So if the decision would be given the output of the plant that needs to reach a certain market size, that it is more economical to go with stainless steel, then still the development of the seating can be done with the single-use design, yet very easily and quickly or seamlessly scale to the stainless steel production bioreactors as well. And overall, this will also help you to mitigate the risks during process transfer. So when considering the different stages of process design, looking at a process development, it is of course critical that we have a small-scale model that accounts for scale effects and that is representative of the proposed commercial design. And this is then where the Amber's 250 automated workstation comes in, which is basically 250 ml bioreactors, 12 to 24 that can be run in parallel, that represent a real bioreactor process. As I showed in the previous slides, the design of these bioreactors is, because of the geometrical similarity, is industrial by design. Here is some data that demonstrates this point, showing the scalability from the Amber's 250 scale to a 5-liter benchtop bioreactors to a 50-liter biostat STR to up to 1,000 liters biostat STR, showing the overlay of yield, viability, and viable cell concentration, which basically shows that the scalability from the 250 ml to the 1,000 liter is definitely achievable, because again, because of the strong geometrical similarity between the different vessels and the different scales. Looking at economics, this is a question that still comes up quite often when we think about Amber's 250. It's a big machine, and of course, customers will very often ask, is it really worth the investment? So of course, there are ways to model that and to really assess the ROI of such an investment in Amber's 250 high throughput workstation. There's different sheets and different ways that we can have those discussions with our customers. These are just a few snapshots of analysis that we have run Here, modeling the hour spent per bioreactor for a traditional 5-liter benchtop bioreactors compared to the hours spent per bioreactor in labor for an AMBER 250 vessel. You see that the consistency in time per bioreactor is pretty much the same, irrespective of how many vessels you operate, and that the time spent per vessel for a 5-liter benchtop bioreactor is about 10 times higher. So that means that you can save approximately 10 times in staff comparing the reusable benchtop design compared to the 250 high throughput options. At the same time, of course, you don't just reduce the time you spend, but at the same time in parallel you will also be increasing the throughput. So this is a graph that shows the number of bioreactor tests that can be run per operator per year. Here again we start with a 6 by 5 mammalian setup, so 6 5-liter benchtop bioreactors in parallel, compared to AMBER 250 vessels. And you see the more vessels you run, the higher of course the number of bioreactor tests that you can run per operator per year, where it shows that you basically comparing the extremes of the scenarios that you can easily increase your throughputs 5x compared to a traditional setup. This is some customer data where we're looking at, again, different options in AMBER 250 versus a 1-liter benchtop bioreactor versus a 2-liter benchtop bioreactor and a 22-liter bioreactor. Looking at different parameters, but I think the one that really stands out here again in the context of time savings to get your molecules through the pipeline quickly is the time to perform 200 runs. And here, if you compare to the 1-liter option or the 2-liter option, you basically can do things 20% or at 20% to 30% of the time with an AMBER 250 48 workstation. Moving on now to process characterization. Also here, AMBER 250 and the geometrical similarity between the scales is very important. So in the process development, the scale-up question is of course critical. In process characterization, the scale-down question will become important. And also here, the similarity in design between the vessels, between the different scales is really critical and a strategic vehicle to make process characterization easier. Especially also when you look at the integration of advanced PAT tools with the AMBER 250 for process characterization purposes, such as integration with the Nova Flax 2, or the integration of single-use sensors with benchtop bioreactors, such as a single-use VIMS sensor or a single-use glucose lactate sensor. This will definitely help to make your AMBER 250 as a powerful workstation for advanced process characterization studies. Now, of course, despite the geometrical similarity from the AMBER 250 to the large-scale stirred tank designs, there's always going to be some inherent physical differences in the different scales, notwithstanding that geometrical similarity. So the challenge is that the small-scale model will very often be able to run over a wider design space than the at-scale model. So when we do process characterization or even process development, we need to make sure that we limit the design space or the parameters, container parameters within the design space that can be accepted by the technology at scale. Very often that's done by limiting the range of the scale-independent parameters such as KLA and power per volume. However, that does not take away the challenge from translating the scale-dependent parameters between scales. So the
hey, Floris?
Yes?
Sorry. The presentation is not showing up on the screen. You might have to do screen share again.
Okay, thank you for that. So I guess I'm not more lucky than you are, Fai.
I do apologize again. If Floris, if you could kindly share your screen again. Thank you.
Thank you for pointing that out. Okay. The screen should be back, right? I assume it is. So the way we at Sartorius have addressed the inherent, notwithstanding the geometrical similarity, the fact that there might still be some inherent physical differences between scales is by integrating the different engineering design spaces of the bioreactors across the different scales in a new software tool. So the point of this tool is to provide a software solution that can help customers to de-risk and expedite the scaling process, both from a scale-down perspective as well as a scale-up perspective. Ultimately, with the goal to have to help customers successfully translate the process between scales, both for PD and as well as PC. So the way this looks is these are a few print screens of the software tool. You would basically start by selecting the scale of the bioreactor and the design of the bioreactor at the starting scale, then the design of the bioreactor at the target scale, selecting some parameters that you want to constrain in your scaling strategy, setting your priorities for scaling, some additional scaling strategic information, and then ultimately the software will give you a scaling strategy, and I recognize it's hard to read, but it will then give you a scaling quality and a scaling overview of the scaling strategy that will help customers then to confidently scale between the different scales. And again, this was built based on the different engineering design spaces and operating parameters of the different bioreactor scales, and combining that with extensive experience in running those bioreactors at different scales for different processes. This is a case study, a very nice case study by Lonza that demonstrates the importance of geometry and the similarity of geometry between scales. I would say the conclusion of the paper is quite interesting, stating that the strategies to match physical chemical conditions are ultimately not sufficient to correct for the differences in geometry, but the right geometry will enable to make sure that also the biological performance can scale along. So I would say that geometrical similarity is an important precondition to also have a strong biological scaling model. So they basically conclude that the geometry between scales is very important. And the way they have demonstrated this is by applying their scale-independent KLA model for their stainless steel stir tank bioreactors that is used at production scale to predict the KLA of two single-use bioreactors. And the two single-use bioreactors have been plotted on this graph. The blue one represents the Sartorius single-use bioreactor, and the red one represents another brand of single-use bioreactor. And what it shows is that because of the strong geometrical similarity in design between the Sartorius single-use STR bioreactor design and the at-scale stainless steel bioreactor design that they use in their manufacturing suites, the correlation is very high between the predicted KLA and the observed KLA. Once again, showing the criticality of having homogeneous and scalable vessel geometries in your PD and PC and manufacturing platforms. Here's a quick pictorial. I know that there's probably more data that there's more things that we can talk about, but this demonstrates data that again comes from customers that show if you start with an amber 250 at small scale for PD and then scale up towards clinical phase over 2 scaling cycles, when you work with a scalable system versus a non-scalable system, that you will be able to generate substantial savings in operational expenditure as well as time to market. So in this scenario, 4 engineering runs were saved because of the fact that the geometrical similarity between those scales could be leveraged. So that means that you save consumables, you save media, you save hours of work. You save resources, but then also having to run less also means that you can get to your desired clinical process faster, which ultimately, again, 4 months is something that is really of significance when we think about the importance of getting biosimilars on the market as soon as possible to be successful. Then moving on from the technology to the software side, what I showed so far is different tools that can be used to generate vast amounts of data that then can be used to confidently scale your process and characterize your process and have a robust manufacturing scale platform. However, of course, your data is only useful if you structure your experiments in the right way and you gather your results in the right way and you analyze your results in such a way that it gives you insightful information about the design space of your process and how to use that design space to build a strong process control strategy. And this is what the software suites, which we call geometric suites, that Sartorius offers, will do. It has different components or different packages. Basically, it has a DOE package, and it has which we call MODI, and it has an NVDA package or a multivariate data analysis package, which we call SIMCA, which will respectively help to structure and run and analyze the results of your experiments in the R&D stage, and then the SIMCA software will help you to build the design space and run a process control strategy against that design space. Tying it all together, technologies and software, it shows you that the Geometrics modules, DOE modules and NVDA modules over the life cycle, and in combination with the different high throughput screening tools and production and characterization tools that I showed with MFCS as a central SCADA, gives you a complete package to gather your data and use your data in an intelligent way to build increasing understanding of your design space and use that design space then to run your process at the desired quality. Looking at some additional platforms to help hit that speed, cost, quality sweet spot when it comes to biosimilar, of course, continuous processing or intensified manufacturing. Time does not permit me today to talk much about the applicability of the single-use stir tank bioreactors that we have for perfusion processes or intensified processes, but the key message is that the package is completely ready and is able to meet all the challenges of continuous or intensified processes at scale. But of course, also it has recently, if you have the at-scale capability, you also need to have the downscale capability, and the Sartorius STR perfusion capabilities has been recently complemented with a perfusion model of the Amber system. So now we also offer the Amber 250 system and the Amber 250 vessels with a perfusion in a perfusion model or in a perfusion setup, where you see that cell retention devices and recirculation loops have been built in, as well as a microspider and two dip tubes to accommodate respectively high cell densities and the recirculation and the exchange of fluids. We talked a lot about upstream, also just a few words about downstream. The Amber platform has also been extended to a UFDF application, which is quite interesting because we move in this way from the complete front end of the process to the complete back end of the process. At first sight, however, what this system allows you to do is also to make sure that early in development you have the tools, high throughput screening tools, to assess different buffers and different formulations in combination with your target protein to make sure that what you effectively want to bring in the market can also be brought on the market in the right formulation, meaning that the proteins will be stable, that you can achieve the right viscosity, right concentration, and the right dosing strategy with your with your biosimilar. And this is something you can do early in the process, often even in combination with your early process development work to make sure that you don't just look at having the right cell lines and having the right expression platforms and development platforms, but also that the cell lines that express the protein can effectively yield the protein that can be formulated in its final dosage form. So as a wrap-up, when we go back to my initial starting points, that for biosimilar strategy or a biosimilar business strategy to be successful, integrated technology platforms are important and will help you to maximize speed, cost, or optimize the optimal point of speed, cost, and quality, we basically presented 3 execution strategies for quick platform development and reliable scale-up to a robust process. First one was a service package, which if you work with a service package that has both cell line development capabilities as well as assay development capabilities, basically provides you with a complete IND-ready package. If you have a cell line that is plug and play, where media and process development has been done, the cell line has been fully characterized, and you can get to research cell bank in 4 months, you basically can get to, after your master cell bank and characterization steps, to a complete IND-ready package in about a year. However, in this talk, in the beginning of the talk, I focused mainly on the analytical testing capabilities that we have, specifically the fully validated set of biosimilar assays, which allow our customers to save substantial time in method development and at the same time safeguarding the CQAs of your products in your process. On the technology side, I presented tools for high throughput screening and quick data collection in the early development side, cell line development on the cell line development side, as well as process development and scale-up side, where the geometrical similarity between the different scales drastically helps to develop robust and scalable processes early in the process. Adding to that, the different layers for PAT and software, for instance, the integration with Nova Flex 2 single-use sensors in the single-use stir-tank bioreactor, and then DOE and NVDA software packages together with MSCS and SCADA, and the scaling tool basically provides you with a complete technology package that will enable a quality by design strategy. And ultimately, QBD by itself is the ultimate framework, I would say, to build quality into your process, and then doing that efficiently by maximizing the technology synergies over the life cycle is the way forward to hit speed and cost in that process as well. Thank you very much.
Thank you so much, Floris. Now we will begin the question and answer segment of the webinar. Once again, I would like to remind the audience that they can still send their questions via the Questions tab located on the right-hand side of the screen. The first question is, what are the other ways to qualify scale-down models? Dr. Poon?
So when I presented, it's kind of a new way to look at the scale down model to qualify it through the metabolism of the cells. But I think traditionally using the KLA, the power number, is also a very good way. So one way, I remember in one of the FDA presentation, they essentially just look at it by comparing the data from the large-scale bioreactor and the small-scale bioreactor. And of course, there's also a statistical analysis you can do, for example, a two-way t-test, just to see if the product quality is equivalent to both the scale. So those are two common ways to qualify your scale-down model. And of course, multivariate analysis is another good way to qualify the scale-down model as well.
Thank you so much, Dr. Poon. The second question is, what are the most important financial drivers in AMBR cost models? Floris?
Thank you, Orsalia. That's a good question. So yeah, I presented a few slides with cost and ROI considerations for the AMBER 250 model. Basically, what it comes down to is that you, because of the high throughput capability, that you save significantly in resources and in labor. It's much quicker to set up. It's much quicker to turn around your vessels, and the time factor will also has a substantial impact on the throughput that you can achieve with your setup. And thirdly, I would say that the footprint is one that is often overlooked, but is also quite critical. the footprint that it takes to set up 1 Ambr 250 in a lab is about 25% to maybe 20% of a similar setup for instance, 24 by 2 liter benchtop bioreactors. So it means that it's easier to set up a lab with less footprint than compared to the traditional way of using benchtop bioreactors. So basically what it comes down to is that you can either do the same with less resources, or you can increase your outputs. You can do more with the same resources that you have today.
Thank you, Floris. Which platform technology is more critical to biosimilar development? Dr. Poon?
So I assume it's the cell line versus media versus the bioreactors. I think for all three, platform technology will be important, because it's a quite commonly asked questions. Usually they say what determines your products the most. So what is more important when you're developing your process? My take is that if you especially with the biosimilar, I think with the cell culture media as well as the bioreactor process, you're actually able to adjust the quality of the product more with these two platforms. And so, but if you have a good cell line to start with, in other words, if your cell lines already have high titer, is already close to the reference product, you actually have less when you're developing, when optimizing a media as well as developing a process. So I would say equivalently important. But I think what people need to focus on is what their expertise is. If their expertise is the process, then focus more on the process. If their expertise in cell line engineering, focus more on the cell line engineering.
Thank you, Dr. Poon. You talked mainly about upstream PD and PC concepts, but what about the manufacturing side? What technology platforms there can drive biosimilar success at scale? Floris.
Right. Yes, I did hint to that a little bit towards the end of my talk, that of course if we want to be successful with biosimilars, also the manufacturing side needs to be considered from a cost of good perspective, and their process intensification is quite important. I did indicate that the single-use stirred tank bioreactors that we have is fully capable and ready for perfusion processes or concentrated fed batch or intensified processes. And we have shown or generated data that show that, for instance, if you work with an N-1 perfusion strategy, now I have to apologize, I don't remember what the associated downstream strategy was, but with the N-1 perfusion, we were able to get the dose cost down to about $50 per gram, which I think is quite competitive. So I think process intensification, either in perfusion or N-1 perfusion or concentrated fat batch, is definitely quite important in order to make sure that you can run at competitive cost of goods. Then, of course, also when you run at scale, the sustainability of your process is quite critical. So I assume that in some cases, or in many cases, I think these days, biosimilar producers or developers will be relying heavily on single-use technology, which makes sense, as I said earlier, of course, to be assessed with a correct and sound economical model behind it. But when single use is being used, of course, your process needs to be sustainable. That means that there needs to be a film in place which is thoroughly controlled by the supplier from a quality and a supply chain perspective to make sure that the film does not turn into a supply chain bottleneck. And at the same time, that the bioreactor or the other operating units are also designed in such a way that they minimize the risk of error and the risk of failure or leakage or operator mistakes. So user friendliness and safeguarding the integrity of the bag by designing the system in a foolproof way, but also providing integrity testing solutions foyer bags before running them, I would say are quite critical. Not always things that people would think of at first when they think about a biosimilar strategy, but certainly important from a sustainability perspective once you have your process ready to go.
Thank you so much, Floris. Before I finish the webinar, I would like to ask our presenters, Dr. Fai, Poon, and Floris de Smet, if they have any closing remarks. Dr. Poon?
I guess my closing remarks is thank you for listening to us, and also I think with biosimilar become more and more common, I think it really is very beneficial to the patient, especially from the cost perspective, and more importantly, as a manufacturer of the biologic or biopharmaceutical company, with the platform technology, I think it really helped to help with the speed to the market. Also, it's important to ensure the safety as well as the efficacy of the drug, so that it will eventually benefit patients.
Thank you so much, Dr. Poon. Floris, do you have any closing remarks?
Also from my side, many thanks to the organizers and Dr. Poon for making me part of this webinar today. Main closing remarks from my side would be really to stress the importance of considering your technology interfaces and synergies between scales when thinking of a strategy to develop and scale up and produce your biosimilars in an efficient way that allow you to really meet those stringent requirements to be successful from a business strategy. So yeah, I think that is quite critical for biosimilars to be successful, not just from a technical or scientific perspective, but also from a business perspective.
Thank you so much, Floris. I would like to take this opportunity to thank our presenters for Dr. H. Fai Poon from QuaCell Biotech Limited and Floris de Smet from Sartorius Stedim Biotech for sharing their knowledge with us. I would like to remind our audience that they can view this webinar on demand by visiting biopharma-asia.com. Thank you, everyone. Have a great day.
Thank you. Bye-bye.
Bye-bye.